I'm from Ontario originally, but I was in New Brunswick for 14 years and moved here a few years ago. So it's lovely to be back in Ontario. I'm going to talk today about the issue of pharmaceuticals in surface waters. It's received a lot of media coverage, so you may be familiar with some of the issues that have arisen. And this is a relatively recent water quality issue. It's not that we've recently started using pharmaceuticals, but it's more recent that we've developed the technologies to measure them in the environment at the very low levels they're present at. So I'll start by talking about where they come from, where they're going in the environment, and then how we can use municipal wastewaters to figure out the illicit drug use that's happening. I'll talk about some of our work on the effects of pharmaceuticals in municipal wastewaters on fish living downstream of the Hamilton and Dundas wastewater treatment plants. And I'll talk about a whole lake experiment that I led, where we added the active ingredient in the pill to a whole lake to look at how it affected the fish population. I'll end by talking about how there's hope for this issue. There's a lot of larger initiatives on the go to reduce our inputs of pharmaceuticals to surface waters. And then I'll certainly be happy to answer any of your questions. There are a number of sources of pharmaceuticals to our surface waters. You can see along the top there we have cattle, we have the pharmaceuticals being used by pets, their disposal, sometimes improperly. They're being used in the agriculture industry. They're released by manufacturing companies, and I'll come back to that later in my talk. But the main route I'm going to talk about today is the toilet and how that's a major source of human pharmaceuticals to surface waters. So wastewater effluents are the largest source of pollution by volume to Canadian waters. And certainly we know there are a lot of contaminants in municipal wastewaters, not just pharmaceuticals. So it's a real mix of industrial chemicals, metals, pesticides, pharmaceuticals, nutrients, organics. But we've been focusing on municipal wastewaters and pharmaceuticals recently because when you and I take pharmaceuticals, we excrete them. They're not completely metabolized in our bodies. We excrete them in our urine and feces and flush them down the toilet into the sewer system. Once they get flushed down the toilet, they go into the sewer system, hopefully into a municipal wastewater treatment plant. But those treatment plants were not designed to remove pharmaceuticals. They were designed to remove nutrients and solids. And so what happens is some of the pharmaceuticals may be lost during the wastewater treatment process, but not all of them. Some of them are broken down quite easily, others are very resistant to the wastewater treatment process, so they can end up being discharged in the wastewater effluents, where they can go into our streams, our rivers, our lakes, our ponds, our wetlands, and have impacts on aquatic species. And I wanted to show a bit of local data. So this is a graph from the Hamilton Wastewater Treatment Plant. These are effluents that were collected by Environment and Climate Change Canada. And they were analyzed for a suite of pharmaceuticals that you can see along the bottom. And they're color-coded by the type of drug. So the green are blood sugar reducers, the blue are painkillers, the red are stomach acid reducers, yellow is anti-seizure medications, the pink, the salmon color, I guess, is heart or cholesterol drugs, allergy drugs, and then there's compounds like caffeine and antibiotics, certainly. So you can see that we find a wide suite of pharmaceuticals in the effluents from this wastewater treatment plant. Basically, it reflects what we use. And I wanted to show you this. There's a very large bar on the left-hand side that actually goes off the scale, and that's a compound called metformin, which is becoming increasingly of concern. And that's because we know now that this compound that's used for type 2 diabetes treatments is actually very resistant to breakdown, and it's present in our rivers at very high levels. And there's some evidence that it affects the ability of fish to reproduce. And we also know that diabetes is increasing in incidence, so the use of metformin will increase as a result. So I talked about using effluents, wastewater effluents, to figure out illicit drug use. And I wanted to show some really interesting work that was done at Trent University by Chris Metcalf. And what they did is they worked backwards. So you can look at the types of drugs that are present in effluents, and then you can back calculate how much is used in the population based on what we know about their breakdown. And then how much a dose is, and ultimately go back to the population that is served by that wastewater treatment plant. So Chris and his team went out to a number of wastewater treatment plants, including the one in Hamilton, to look at the types of illicit drugs and some prescription drugs that are used in those populations. So this is a list of all the different drugs they measured, things like cocaine, codeine, different types of amphetamines. So they went to 4 different treatment plants in this study, so Peterborough, Brantford, Hamilton, and Montreal. And this is a table that shows you the number of people that are served by those wastewater treatment plants, as well as the type of treatment. So some of it was more advanced treatment than others. And they collected wastewater effluents and measured them for these illicit and prescription drugs. And I'll just show you, I hope you can read that, or maybe you can't, but these are the levels of different drugs in the wastewater treatment plant effluent. And what this graph is showing you is some of the different compounds that they found in the effluents during the week in Montreal, which is the first group of bars, and then on the weekend in Montreal, you can see this huge spike in cocaine. And here's Hamilton in the middle group of bars on the weekday, and then on the weekend, again, we too have this big spike in cocaine. And then when you look at Peterborough, they don't have that spike in cocaine on the weekend. So it's interesting. To me, it's really interesting that we could start using these kinds of approaches to figure out cocaine use, for example, in our population. So my work really focuses on what happens in the aquatic environment when pharmaceuticals are released to our rivers and lakes. And we know from a lot of the chemistry that's been done that fish are exposed to hundreds of different types of pharmaceuticals. And I've listed some of the different categories that we've measured in surface waters here. So there's a really diverse suite of pharmaceuticals that are found in our rivers, including downstream of the Hamilton and Dundas wastewater treatment plants. And what happens to some of those drugs? Well, some of them will accumulate in fish tissues. So far we've found almost 90 different types of pharmaceuticals in the blood or the flesh or the brain of fish. Some of them will accumulate in the food of the fish. And what's really sobering for me, and something that's relatively new in this area, is that you can actually get the movement of pharmaceuticals from aquatic systems to terrestrial systems as the insects that live in those streams emerge. So they'll start as larvae, and then they'll transform to an adult and fly out of the stream, and with that they'll carry some of the pharmaceuticals in their tissues. And what happens then is that they become a good food source for bats and birds, and those pharmaceuticals, and spiders, and those pharmaceuticals get transferred to those predators. So there's a real concern and a big unknown here about what's happening in the terrestrial ecosystems around wastewater treatment plants. What does it mean for them to have a diet with amphetamines or blood thinners, or you name it, different types of pharmaceuticals in their diet. So fish are exposed to pharmaceuticals mainly through water, so they take them up in their gills, or they can also consume them in their diet when the pharmaceuticals accumulate in their prey. And then sometimes they're excreted quickly, sometimes they're accumulated in the tissues. It really depends on the type of pharmaceutical, the chemical characteristics of the pharmaceutical itself. And we use pharmaceuticals because we know they're biologically active and effective in our bodies. But maybe you didn't know that a lot of the reactions that they have in our bodies can be quite similar to the reactions that they have in fish and other wildlife and other aquatic species. And that's because pharmaceuticals react through a receptor. It's like a lock, and the drug is like a key. And oftentimes those locks are present in not just in humans, but also in fish and many other species. So when other non-human species are exposed to pharmaceuticals, they can have very similar effects in those organisms as they do in our bodies. Is everyone understanding that? Great. So we know that they can be biologically active in fish and other life. Sometimes they have similar effects, sometimes they surprise us and have unpredictable effects. So fish are getting exposed to a suite of pharmaceuticals. The blood lipid regulators actually reduce blood lipid levels in fish. Diabetes medications affect fish reproduction. Antidepressants alter our behaviors, and they also alter behaviors in fish. But surprisingly, they affect the ability of clams to reproduce, or the timing of clam reproduction. We also have another surprise is antibiotics, when they're present in our surface waters, can interfere with plants and their ability to photosynthesize. And the last one I mentioned here is anti-inflammatories can impact the growth of fish as well. So there's a suite of pharmaceuticals out there and a suite of effects that they're having on our fish and other aquatic life. I wanted to mention some local research that's been going on for a number of years, well before I started at McMaster. And it's being headed up by Sigal Balshine, and she's in the psychology department. And it's myself and Bram Scott in the biology department that are working together in this large collaborative effort. We're focusing on both the Hamilton Wastewater Treatment Plant and the Dundas Wastewater Treatment Plant, and over several years we've been looking at fish communities there and the response of those fish to the effluents. So we've sampled a number of locations. Some of them are very close to where the wastewater is being discharged, some of them are further away, with the belief that as you move away from the source, the exposures and the effects of those contaminants will be reduced. So to show you some of the work we've been doing is measuring different types of pharmaceuticals in the Desjardins Canal, for example, or downstream of the Woodward treatment plant. And these are some of the drugs that we see right where the Dundas outfall is discharged, which is on the far left of the graph, and typically the very high values in the bars. And then as you move away from the outfall, the levels of these pharmaceuticals in the water goes down. And here we're showing painkillers, different types of anti-inflammatories, antibiotics, antidepressants, beta blockers, and then some measures of diet, caffeine and sucralose, for example. And this is very typical. When you look right at a wastewater treatment outfall, you see very high levels of pharmaceuticals that get diluted as you move downstream. So certainly the risks to any fish in that area are greatest for ones that are living right where the discharge is happening. So we're out there collecting different fish. We focus on round goby, which is an invasive species in this area, and then two different types of sunfish, the green sunfish and the bluegill sunfish. And these are quite common in this area as well. We'll go out typically in the summer, but also in the winter over the last couple of years and net the fish. So put out these traps like you see in the picture here. We also do some electrofishing, which is basically putting a current into the water which stuns the fish and causes them to float up to the top. We catch them that way, or we use some seine nets, which are basically like big purses that you drag through the water. And then we bring the fish we're interested in back to the lab and look at different measures of health. We've also measured their size, and size is an indication of how well they're eating. And these are some of the masses or weights of fish from the different sites. So the green are the Woodward site, the far left bar is the site with the outfall, and then the far right site is the clean site for the sunfish. And then the middle two bars are the bluegill sunfish, and then the far right red bars are the round goby. And the main message from this graph is that fish caught right near the effluent outfalls tend to be fatter. And that is probably because there's more food. It's warmer, they're attracted to that kind of environment. And so they tend to feed more and grow better in those environments. But what we also find is when we put them in cages and keep them in the cage for a number of days, that the fish that are in the cage near the wastewater treatment plant, which is the bottom line on the graph, don't survive as well as the fish that are caged in areas away from the outfall. So they're clearly getting exposed to something in the wastewater treatment plant effluent that's not allowing them to thrive in that environment. But I also wanted to touch on a whole lake experiment that I led it started when I was working with Fisheries and Oceans Canada, and then has continued for a number of years after that. And I'll talk a bit about the motivation behind adding the pill to a whole lake, and it's also related to pharmaceuticals and wastewater effluents. And it goes back to the 1990s. So researchers in the UK were finding very high levels of feminized male fish in their rivers. In some of the rivers in the UK, all of the males were showing signs of feminization. And obviously that was causing a lot of concern and a lot of investigations into what was causing those male fish to become feminized. And initially, there was a lot of detective work, and there was some thinking that maybe it was detergents that were causing these male fish to have smaller gonads, produce eggs in their testes, as well as egg yolk proteins. Maybe it was pesticides, maybe it was industrial chemicals, but in the end, we pointed the finger at the presence of estrogens in the wastewater. So these are natural estrogens that women and men excrete, but also the synthetic estrogens that are used for hormone replacement therapy as well as birth control. And that soup of estrogens was getting taken up by the gills of the male fish, and it was causing them to develop eggs in the more severe cases. And male fish with eggs raised a lot of alarm bells. It motivated a lot of studies worldwide, and certainly there have been a number of researchers in Canada that have looked for feminized male fish in our rivers. Do we have them? Yes, we do. So we have them in the South Saskatchewan River. We have them in the St. Lawrence River downstream of Montreal. We have them in the Grand River downstream of Kitchener and Waterloo. And the common theme here is these are all small fish. And small fish tend to stay in a very local area. So they don't move a lot in the rivers and lakes. So they'll stay in one area, their exposure to estrogens is higher. But we also tend to find them where wastewater treatment is poor and where human populations are high. So it's a combination of the type of fish, the type of treatment, and then also whether there's any dilution of the effluents. So we have feminized male fish. I wanted to show you what it looks like inside. So the picture on the right is of a darter. It's a beautiful fish fairly small. It's very widespread in the Grand River in southern Ontario. And when you open up the male fish, you see some white tissues inside. Those are the testes, the sperm, right before spawning, or right before reproduction. And what Mark Servos and his group at Waterloo found very obviously was the presence of eggs. So sometimes when you open up the fish and you look inside, you can see these round yellow objects, and those are eggs that the fish has produced because of the exposure to estrogens. You can also look at the tissues under a microscope, which is the picture on your right, and you can see the round objects are the eggs, and around them are sperm cells. And Mark's team has found that in some of the parts of the Grand River, over half of the males have this very severe intersex condition. So we knew there were feminized males out there. We knew that estrogens were causing the problem. We wanted to do a whole lake experiment, and we did this at a fantastic research facility called the Experimental Lakes Area. Has anyone heard of the Experimental Lakes Area before? A few of you, great. Well, for those of you that haven't, I'm pleased to be introducing it to you today because it's a jewel of Canadian science, and it's really well known internationally for its whole lake experiments and for the impact that those experiments have had on regulations. And I'll mention a few examples of that shortly, but it was set up in the late 60s. It was a federal government facility for a long time, and it is a series of lakes that have been set aside for experiments. So there's no commercial fishing, there's no sports fishing, there's no development in the watersheds of these lakes. They're set aside specifically for research. In 2012, the federal government cut the funding for this facility. It was in the news. It raised a lot of media attention because of the facility is unique in the world. And lucky for us, the International Institute of Sustainable Development came along and took over the facility, and it's doing really well. It's thriving under their control, under their management. So I call it a zombie research station because it's come back to life after the federal government cut the funding. So this is a map of some of the lakes that are in the Experimental Lakes area. There's a great field station you'll see a picture of shortly, and a number of lakes that have been used for whole lake experiments. So there's some examples here of lakes for acidification studies, for nutrient studies, for estrogen studies, which I'll talk about shortly. There's one on mercury, there's one on aquaculture, et cetera, et cetera. And the two experiments that had the biggest impact on our legislation were the ones looking at phosphates in detergents. So when you look at your detergent bottle and it says phosphate-free, that's because of the experiments at the Experimental Lakes area. So those experiments were very influential at reducing phosphates in detergents and improving wastewater treatment. And then the other one that's really had a lot of impact is the acidification studies. And so the acid rain problems of the 80s and 90s were largely solved because of some of the studies that were done at the Experimental Lakes area. So these are some pictures. You see the top left is an aerial photograph of some of the lakes, so there's no development in the watershed. And then it's got a year-round facility with labs and residences, so it has up to 70 people at the camp full-time in the summers. And really this is a fantastic facility, very high-impact science that has influenced a lot of legislation, regulations, environmental policies, and that's because it's designed to answer questions that you can't answer anywhere else. So we decided to add the pill to a whole lake for a few reasons. We knew that estrogens in municipal wastewaters were feminizing male fish. But the big question that we wanted to answer was the so what question. So those males with eggs in them, can they still successfully reproduce, or is there going to be impacts on the numbers of fish, the sustainability of the fish populations? So that was our million-dollar question. We chose a lake at the ELA to do this experiment, and it's quite a large lake. It has one small lake upstream and a very big lake downstream. It's big enough to have trout in it, so it's 14 meters deep. And what we did was we did some before studies, during studies, and after studies. So we spent a couple of years looking at the lake in its natural state. And this is the beauty of whole lake experiments at this facility, is you can get a sense of the status of the lake before you manipulate it. And that's really unique, because usually we're looking at things after the fact. And then for 3 summers in the early 2000s, we were adding very low amounts of the estrogen that's used in the birth control pill. And I'll show you shortly that as soon as we started adding it, it had profound impacts on the fish living in that lake. And then we spent a number of years after studying the recovery of the system. And what we were adding is about 10,500 pills worth of this synthetic estrogen a day, which when you look at the demographics of the pill use in Canada, that equates to what's being used in a city of about 240,000 people. We had to add it 3 times a week because sunlight and bacteria break it down, so it's not persistent in the water. And then certainly we were measuring it to make sure we were getting our target levels. I'll talk only about what happened to this fish called the Fathead minnow, but certainly we were looking at everything living in the lake too. And we focused on the Fathead minnow because it's a short-lived species. It's a very common food source for big fish like trout, and it tends to reproduce only once in its lifetime in the middle of summer. And we caught it mostly with minnow traps, but also with the trap net you see in the top left. So what happened to the fish? This is a graph of vitellogenin levels in the male fish. And vitellogenin is an egg yolk protein that you normally see when females are developing their eggs. But what happens for males is when they take it up through their gills, it causes their liver cells to also produce this protein. So it's used as a measure of estrogen exposure in the environment in male fish. And the graph is showing our experimental lake in blue and then our 2 reference lakes in gray and green before and during the estrogen additions. And you can see that first really large bar there. The males, when they took the estrogen into their bodies, responded by producing very high levels of this egg yolk protein. And that continued through all the summers of our estrogen additions. Then we were looking at their ability to develop their sperm cells, and this is a cross-section of their testes or gonads. And this is what a normal male would look like. Those little dots in there are all sperm cells right before the spawning season or the reproductive season. That's normal. And then the next picture is a male that was exposed to estrogens for 1 summer. And you can see just on a broad sense that the testes looked very different. They lost their ability to develop their sperm cells. The tissues were very chaotic, disorganized. There was a lot of interference of the estrogen with their ability to reproduce. And then what we saw after 3 years was this intersex condition I talked about earlier from the UK studies where the males started producing eggs. So those round objects you see on the slide are eggs in amongst the sperm cells. So what happened to the population? We also looked at the numbers of fish, and this graph here is the numbers of young fathead minnow before and during the estrogen exposures. And the black symbols, those are from our experimental lake, and what we saw was a reproductive failure. So the fish could not reproduce and their numbers crashed. The numbers of young crashed. And then the adults, their numbers were sustained for a while during the exposures, and then shortly after, their numbers also crashed by about 99%. So because of this additions, we saw a near extinction of the fathead minnow from the lake. And that went back to answer our million-dollar question. Well, can those fish still successfully reproduce, or is there going to be an impact on the sustainability of that population? And yes, indeed, there was a very severe reduction in fish numbers because of the estrogens. So if you look at this from a Canadian population perspective, this is a bit old, but if we had around 37 million and we lost 99% of our population, we'd be down to 370,000. So that's just gives you a sense of how dramatic this response was for the fathead minnow. And why did they stop reproducing? Well, we don't really know, but it was a combination of things. So maybe you were wondering why fathead minnows are called fathead minnows. It's kind of a strange name. But the top fish there is a male fathead during the reproductive season. And what they do is they develop dark banding, they get a pad on their head, which is why they're called fatheads, and they get a bunch of bumps on their nose. And I guess all of this makes them very sexy to the female fathead minnow. And what happened once we started adding the estrogen is that the males stopped developing the coloring in the bumpy heads, and they lost all that external maleness, and became much more like the females. So I suspect there was a lot of confusion around who was male, who was female. But they also started behaving differently. They were laying fewer eggs. There was a number of things that were going on as a result of the estrogen exposures. So was there any recovery? And I love talking about this part because it's good news. We stopped adding the estrogen after the 3rd summer. And what we saw, this was the graph you saw before with the high ADO protein levels during the estrogen additions, and then the levels came back down to their previous low levels after the additions were stopped. These were the slides you saw, the microscope slides with the male tissues showing the eggs. And this is what we saw 4 years later, was that they recovered their ability to develop their sperm cells. And then this last slide, I wish I had I'm not very technically savvy, but if I was, I would put in some drum rolls or maybe the music to Rocky as he's running up the stairs or something. But what we also saw in our catches here was a complete recovery of the fathead minnow in one of the springs. So there must have been great mood music this summer before, and they just reproduced like crazy, and their numbers came back up to what they were before we started our experiment. So it was excellent news and quite a surprise. So just to summarize what we learned from the experiment, this is a picture representation that shows all of the different fish and other non-fish species that we looked at. So we've not only looked at the fish, but what's living in the water column and what's living in the mud in the shallows of the lake. And certainly the biggest response was in the fathead minnow. Its populations went down, but we also saw some decreases in other fish as well, like the sculpin and the lake trout. And then because of that, the predators disappeared, the prey went yahoo, nobody's around to eat us, so they increased in abundance. So some of the insects went up in numbers. So just to summarize, we saw direct effects on the fish population, which was our million-dollar question. We also saw indirect effects on the lake trout, which I am happy to explain later. And we saw some changes in the prey. And the most important lesson from this study was that whole lake experiments are invaluable. because you could never repeat this kind of study in the lab. It's impossible to have all of those organisms together to understand how they're responding directly, but also indirectly when their predators and prey change. So I'm often asked, well, does this mean that women should stop taking the birth control pill? And it's a mix of good news and bad news from our studies. So the bad news is that certainly you can affect fish populations when you have estrogens in the municipal wastewaters. And the pill is as effective a form of contraception for fish as it is for humans. And we can also see indirect effects. So when you reduce numbers of fish, that means you have knock-on effects on other organisms living in the food web. But the good news is that when you remove that source, that exposure, you can have recovery. And that's what we saw when we stopped adding the EE2 to the lake. And what I'm really excited about, and I'll talk a bit more about what's happening in Canada around municipal wastewater treatment, is that we're starting to see wastewaters not as a waste, but as a resource. And it's a resource of energy, it's a resource of fertilizer, the phosphorus that we need for agriculture. And I think the tides are changing in that we're not just focused on treating it to reduce its impacts on surface waters. We're starting to consider it as a source as a resource instead, I guess. So the philosophy is changing around wastewaters. And I think that will have implications for this whole issue around pharmaceuticals, because as we recover energy from our wastewaters and phosphorus, we're going to reduce our impacts, our inputs of pharmaceuticals in the effluents. So should women stop taking the birth control pill? And my unequivocal answer is no. The answer is in better wastewater treatment. And so as we move from lower to higher treatments, from primary to advanced treatments, we can remove a lot of the estrogens that are present in the wastewaters, up to 97% with advanced treatments. So that's really where the answer lies, is to make sure that we have as much treatment of these effluents as we can. And I wanted to show you a bit of information on wastewater treatment in Canada. So this is information from Statistics Canada from 2017. And there's information for Canada on the top about the relative proportions of different types of treatment for wastewater effluents, and then there's all the different provinces listed below that. The light blue bars are showing you the proportion of effluents that are treated only to primary levels, and that's just screening of solids. The dark blue bars are proportions of secondary treatment, and that's when we bring in bacteria and oxygen and mix it up, and it reduces a lot of the organics that are present. And then we have the red, which is advanced treatment, and the gray, which is no treatment. So you can see in Ontario here that we treat our effluents to a very high standard. And that's not the case across all provinces and territories in Canada, but the tide is changing, and I'll explain that. The good news is that we have new regulations that are in force now under the Fisheries Act, so they're wastewater effluent systems regulations, wastewater systems effluent regulations, and so they came into force in 2015. And there, as a result of those regulations, a lot of wastewater treatment plants are upgrading their processes to meet the requirements of the regulations. And so here in Ontario alone, we've invested almost $3.9 billion in upgrading our systems. So those upgrades will have benefits for the fish living downstream. And here's an example of the benefits. So this is the Kitchener wastewater treatment plant, which discharges into the Grand River. And the Grand River is where we see all of those high intersex fish, a lot of feminized male fish living in that system. And all of these bars here on the graph represent ammonia levels. And ammonia is a compound that can be quite toxic to fish. And the plant upgraded to a new system to remove ammonia. So what you see is higher ammonia levels, and then as the upgrade happened, the ammonia levels dropped. When they did those upgrades for ammonia, their systems also improved the treatment of pharmaceuticals. So the number of pharmaceuticals dropped, as well as the levels of pharmaceuticals in the effluents. So that was a byproduct of the upgrade. It wasn't upgraded to reduce pharmaceuticals, it was upgraded to reduce ammonia, but it had a pleasant and much appreciated benefit of reducing pharmaceuticals at the same time. So what happened to the fish? I told you that there's a lot of intersex in the male darters living in the system. And the blue bars here, the first group of blue bars, those are fish from upstream of the Kitchener wastewater treatment plant. The middle group of bars are from downstream of the discharge, and then there's another group of bars on the far right that are further downstream. And the red, if you just look at the red bars, you'll see that as soon as the wastewater treatment plant upgrades happened, the fish that had this intersex condition dropped over several years. So it went down quite dramatically within 3 years of the treatment upgrades. So this is great news that when you reduce the estrogens, then the fish respond by reducing the intersex condition. So the last bit I wanted to mention here was manufacturing, because this is an emerging issue and something that we're looking at in Canada. There's pharmaceutical manufacturers worldwide, and they're producing all of the drugs that we're using. And as they produce those drugs, they're releasing the wastes. So those plants are coming under greater scrutiny. And that's because there's been studies. It started in India, where they looked at the pharmaceutical company. They took water from downstream of where the company was discharging, and they found astronomically high levels of antibiotics in the surface waters, like millions of times higher than anything we would see in Canadian waters. And this generated a ton of concern, not only because it could impact aquatic life living in these systems, but also because of the whole issue around antibiotic resistance. So what happens when you put antibiotics into a river and it mixes with the bacteria that are there? Are you creating the conditions to cause more antibiotic resistance that could then get transferred to humans or wildlife? So that's something that's causing a lot of concern, and certainly something that's being investigated in Canada. The last message I wanted to leave you with is for something you can do to improve the situation is take your unused medications back to pharmacies. And there's a group called the Health Products Stewardship Association that works with pharmacies to properly dispose of all of the medications. So you can return your medications to a number of pharmacies in the Hamilton area, and they'll go to an incinerator, which is much safer than flushing them down the toilet or putting them in the garbage to go to the landfill. There has been some media coverage about pharmaceuticals in drinking water, so I wanted to touch on it as well in case you were concerned about that. There have been a number of studies in Canada measuring pharmaceuticals in drinking water, and sometimes they do find different types of drugs in our drinking water. It just depends on the source of the drinking water. Is it downstream of a wastewater treatment plant, or is it not treated with advanced ozonation, for example? But the levels tend to be very low. Some of the ones that they're finding are some antibiotics, like I always have trouble pronouncing these names, oxafloxacin, carbamazepine, which is an anti-epileptic drug, ibuprofen, and then gemfibrozil, which is a lipid regulator. And those tend to be more common in drinking water, but you can also find some other pharmaceuticals, some other antibiotics. But they've done a risk assessment, and it would be impossible for anyone to drink enough drinking water with pharmaceuticals in it to get what we would take in a dose. Physically impossible to ingest that much through drinking water, and we know that pharmaceutical companies do a lot of testing before products come on the market to make sure that they're safe for use. Pharmaceuticals are designed to have biological effects and therapeutic effects in our bodies, and it's been a rude awakening to find that they also can have effects in other species. We know that there's very diverse and numerous types of pharmaceuticals that are being found in our surface waters, and that they are having effects on fish and other aquatic life, and they're getting transferred to spiders and birds that are feeding on those insects that are emerging. So there are concerns in Canada and globally around the presence of these contaminants in our surface waters. We're only able right now to measure a small fraction of what we actually use because we don't have the analytical abilities to measure the 4,000 that we know are in our market. So the question is, what are those other pharmaceuticals, what are the impacts those other pharmaceuticals are having on our aquatic life? There's lots that's unknown still, so it's a good thing I have some of my career left to look at these issues. But the last point, and the most important one, I think, is that to improve this issue, we're going to have a collaborative effort between manufacturers, consumers, and governments to really ensure that pharmaceutical use is sustainable. So proper disposal of the wastes from manufacturing companies, proper disposal of unused medications, reducing the use of unnecessary medications, and also improving the design of pharmaceuticals so that they're more easily broken down when they're in the environment. So adopting some green chemistry principles, I think, is very needed in this issue. There have been a lot of supporters of our work over the years, and I've listed some here. Our local work was supported by RBC, the Royal Botanical Gardens, and the City of Hamilton, as well as Fisheries and Oceans Canada. So I'll leave the rest to your reading, and thank you very much for your attention.